Hello and welcome to today's GenQA Focus On webinar, whereby we are absolutely delighted to be joined today by our GenQA consultant, Dr. Ros Hastings, who is the standing chair of the ISCN committee and is here to give an expert review of the changes of the ISCN for 2024 and give you some worked examples, and then we're going to have a quiz to see if you can pick up some of the changes that have happened through the changes, the update to the nomenclature guidelines. I'm Professor Sandy Deans and I'm Director of GenQA, and I just thought I'd start this opportunity with a few slides giving some background about GenQA, and then I'll hand over to Ros. We are very happy to take some questions as well, so if you would like to submit a question, please do so using the questions function that hopefully you can see on your screen as part of the menu. So next slide please, Emma. So hopefully you have all heard and know about GenQA, but GenQA is part of the UK NEQAS consortium. So this is the UK National External Quality Assessment Service. And really what the services are about are to make sure that the clinical laboratory test results, they're accurate, they're reliable and comparable wherever they're produced. So for example, if one patient was tested in a laboratory in Edinburgh, say in the GenQA lab, the same result and the same clinical interpretation would be provided by another laboratory based somewhere else in the world. UK NEQAS itself is a charity organisation, and the central office is based in Sheffield in the UK, and all of the NEQAS schemes are self-funding, not profit-making schemes, which means we charge participation fees, but any surplus at year end, and we do try to keep the costs as low as possible, are fed back into the scheme so we can generate and develop new pilot EQAs, which is really pertinent for the genomics world because we're constantly being asked to set up new external quality assessment schemes. UK NEQAS is comprised of about 20 specialist centres. They're all dotted across the UK, and really they've evolved from individuals' expertise in particular areas of laboratory medicine and an interest in quality. So there's a UK NEQAS for microbiology, one for clinical chemistry, one for haematology. So GenQA is the UK NEQAS centre that delivers all of the EQAs for genomics. UK NEQAS has been around for over 50 years, and despite being called UK NEQAS, we actually have participants worldwide. And in fact, in GenQA, I would say about 80 to 85% of our laboratories are actually based out with the UK. All of the UK NEQAS schemes are also ISO accredited, so we feel the pain of that as the labs do, and there is an independent standard for external quality assessment providers. Next slide, please, Emma. This is just a screenshot just to show some of our stats. So this is the latest off our website. We have about 2,500 participating laboratories or centres. We are absolutely delighted to have over 200 different expert advisors, and I think that's something that we all enjoy at GenQA, is working with experts in our field across all of our different specialties that we provide assessments for. For 2025, we have 126 EQAs being offered, and some of those have multiple distributions throughout the year, and we now have 101 different participating countries. Next slide, please. I think our unique selling point is that we cover assessments from the end to end of all of the genomic pathway. So that's right for our clinical colleagues and our genetic counsellors, the pre-test and consultation and the referral process, right through to how you handle your samples, so pre-analytical assessments, the actual extraction of DNA, and we're moving into RNA, the quality and the quantity and the quantification of that, then the testing accuracy, and that's for a whole remit of different specialties, as I mentioned. And then more and more challenging is the interpretation of the genomic data that is generated, and then the safe reporting, and that's where standardised nomenclature comes into its own. Next slide, please. So it's now my great pleasure to hand over to my colleague Ros, who is going to talk about implementation of ISCN 2024 and how GenQA has been involved in that and supporting our participants. So Ros, over to you, please.
Thank you, Sandy. Can I have the next slide, Jen, please? So just to say there's potentially a conflict of interest. GenQA supports ISCN in all its EQAs where it's relevant to use in nomenclature, and I also the ISCN Standing Committee Chair. Some may perceive this as a conflict of interest, but being as GenQA supports the use of ISCN, personally, I don't think there is. Can I have the next slide, please? So I'm going to talk about the ISCN background, why it's needed, nomenclature, educational tools that we have through GenQA, and what's changed, what the changes are for ISCN 2024, and then we've got the quiz. So hopefully we'll get through all the questions, but it will all depend on how quickly I get through the slides. So let's have the next slide, please. So just to give you a bit of history, initially the first publication of the nomenclature was in 1971. The first time it included neoplasia wasn't until 1991, and each edition has included lots of different revisions. And the biggest revision actually is in 2024, where we've revised the structure, we've codified it, we've included genome mapping and lots more examples because the complexity of the reporting we are now doing. Next slide, please. So why do we need nomenclature? It's a requirement for accurate reporting results and the interpretation by clinicians. And it does mean that patients' results can be transferable across countries without any language barrier and can be understood by the different healthcare providers. So it's important we actually get the nomenclature correct. Otherwise, we do get confusion about what the results mean. And it helps in the advancement of knowledge through communication of results in publications, plus also database entries. Next slide, please. With the changes in the way that we do our technologies, there is now much more of an overlap between ISCN and HGBS. And so basically ISCN is used to describe variants at a chromosome gene level, not at the nucleotide amino acid level. And it includes numerical changes, structural abnormalities, pathogenic variants, haplotypes, repeat expansions, and you have things like duplications, deletions, inversions, insertions, translocations. And what you do is you describe what you actually see. So if you've got a trisomy 21 as a simple example, then it'd be ×3. With HGBS, it's used to describe DNA, RNA, protein sequence variants at the gene, nucleotide, and amino acid level. And it's that gene level where we've got an overlap. And usually these are frame shift, missense, nonsense, insertion, deletion, duplication, tandem repeats, and SBs. What it does do, HGBS, it describes a change in one chromosome or the structure of the chromosome compared to a reference sequence. So if you had trisomy 21, it'd be ×2 because you've got an additional copy. And so that is a fundamental difference between the two nomenclatures. And we are trying to work close together. We're both on each other's committees, but there's still some work to do. Can I have the next slide, please? So ISCN covers karyotypes, FISH, region-specific abnormalities, arrays, optical genome mapping, and sequencing. And that's the important part to stress. Can I have the next slide, please? So we've restructured the ISCN, and the general rules are now summarised in Chapter 4. We've got specific technique rules in each of the individual chapters. To make life easier for the diagnostic labs, we've included the ISCN examples are all got alphanumeric numbers, and the rules are all alphabetically numbered. In additional nomenclature examples have been included, some of them are more complex, and we've expanded the regional specific assay section. So it now includes targeted chromosome analysis, which I will describe, targeted and microarray analysis, because some people are only looking at a specific region, fusion genes, expansion repeats, and methylation assays for imprinting. And there's a new chapter on genome mapping. And if you have got the book and you wonder where the ideograms have gone, they've gone to the appendix. If you haven't bought the book, can I strongly recommend you do? You get online access for the whole period of the life cycle of that particular book. And if you haven't bought it, I've heard from my other European colleagues that it's actually cheaper to go via Amazon than going via cargo. We've used the genome build GRCH38 throughout. for all the cytogenomic techniques, and we've tried to streamline the nomenclature so it's now consistent across the different technologies. Can I have the next slide, please? So the changes, the predominant big changes are those in red. All the others are relatively similar, although there obviously are new examples, and we've tried to condense it now so there are 16 chapters. It is a much bigger book, and hopefully once you've got used to it and you've read it through, you will understand why we've made the changes that we have made. So the general rules now is, because they were all over the place, they're now incorporated into, they've incorporated chapters 4, 5, 6 and part of 8, and the karyotype chapter, which is now in ISCN 24, incorporates the rest of chapter 8 and chapter 5. Can I have the next slide, please? So this is just a quick graph to show you where all the different examples are, and obviously genome mapping is a new one, so there's a lot of examples there, but neoplasia, RSA, sequencing, all have got additional examples as across the whole of the different chapters. Next slide, please. However, when we actually did this, we did actually, unfortunately, have some errors. Some of them were not picked up in proofreading, others were introduced by the publishers. We have now got 13 ISCN errors identified in the book. There is a list on the ISCN online forum, so you can do that, and it is corrected on the online version. All the corrections are published in cytogenetic and genomic research, predominantly in the fish section, but there is one in chapter 4, a couple in chapter 6, and one in 8 and one in chapter 11. They're predominantly to breakpoints or incomplete ISCN or rule errors or punctuation errors. Next slide, please. So we've got plenty of educational resources which we can now use to help us to get used to the new version. So you've got the book, you've got the online version, and you've got the forum, which I'll just show a few pictures of in a minute. You've also got the EQAs offered through GenQA. Or if you come from another country, sometimes it's referred to as proficiency testing. Now, there are several national and international EQA providers that include ISCN in their assessments. Currently, GenQA has a specific ISCN EQA. There are publications, there's a summary of all the changes in cytogenetic and genome research, and we published a preliminary document on genome mapping in the same journal. There are also, which I'm going to discuss, the individual competency tools, which are modular, which have been released for both ISCN and HGVS. Plus, there are webinars like the one we're now doing for GenQA. There's going to be one in the summer with some complex cases for the ASHG. And there's a series that have been done for Cancer Genomics Consortium. Plus, every now and again, GenQA offers ISCN workshops. Can I have the next slide, please? So if you go online, this is what you will see. It's a totally different picture. That's the website address. You have to sign in. Next slide, please. And you can see from this slide that there is a forum section, and you can either go through the top bar or you can go individually, or you can, and if you want to go to a specific ISCN, you go to the table of contents and you can do a search, so you can look for things. So it makes it a bit easier than perhaps trawling through the book. Can I have the next slide, please? And you can put your queries on the forum. You can look at previous ones and read what all the responses are. Next slide, please. So I mentioned about Genie, so I'm just going to talk a bit about that. Can I have the next slide, please? So this is an individual online training and competency tool. It is for individuals, not for laboratories. So obviously the laboratories can subscribe for the whole of the laboratory, but it's an individual-based competency and training tool. It's modular and it focuses on areas of genomics where the evidence of competence is needed. I say you can pay for it individually for a year, or you can do it as a laboratory, which is for 10 users, and obviously is cheaper if you could do it as a laboratory. And the website address is at the bottom there. Next slide, please. So this Genie has module for all the different competencies, and you can see them all at the bottom. There's various different ones have now been used by over 8,000 individuals, and that was the latest figure in June of this year. And in the next slide, please, you can see that there are educational videos on extraction and laboratory tours. There's some on variants. There's some on constitutional, on ISCN, including constitutional neoplasia karyotyping, but FISH and others are coming on board as well, and HGVS. And there's some on data analysis where you've got proportionate analysis of the whole genome or exome sequencing data. Also to mention is that this module, modular system Genie, is now CPD certificate certified. Now the next slide, please. So if you register, if you go to the HGVS module, then you will get information about what the reference sequence is, what the deletion is, and then you will select a series of possibly 10 options and choose the right one. And for the ISCN, which is on the next slide. Next slide, please. Then it's similar. You get the information about what the actual abnormality is. You get a picture of it, so you can actually visualize it, and then you choose from a selection of abnormal results which one is the actual correct ISCN. Next slide, please. So the educational videos, we've now got analysis of CMVs and SMVs from NGS data. For the variants, we've got somatic variants for solid tumours and also for hematological malignancies. For the nomenclature, we've got intermediate, advanced, as well as basic levels. And for the data analysis, there's risk calculations, linkage analysis, karyotyping, and phenotyping. Can I have the next slide, please? And just to show you where we're coming with the ISCN, the Constitutional Intermediate Advanced will be released in August. And then following this will be a microarrays, basic intermediate, FISH intermediate, advanced, neoplasia karyotyping, neoplasia microarrays, regional specific ones, sequencing and optical genome mapping. So it's an ongoing program and it will be expanded on an ongoing basis. And just to show you what some of the results have been like, can I have the next slide, please? So this is the ISCN, the full module results for constitutional basic and neoplasia basic, and there were about 38, 37 cases. Less people did the neoplasia, but it hadn't been open for as long. And the constitutional, we had 250 participants. Of the number of cases that were analysed, you can see there's quite a few there because obviously each person gets 5 randomly assigned cases. So even if you're working in the same laboratory, you are very unlikely to get exactly the same 5 cases. So you can't cross and compare. Unfortunately, although it's a high figure, I would love to have seen it much higher for a basic level. Only about 74–75% of the results that were submitted were correct. And there were some that fortunately went back and tried to repeat and tried to improve. But there's only a small number. And you can keep going back in as an educational tool and as a training tool so that you can learn from your mistakes, or that you may get a different answer. I'm going to explain to you what happens when you do get your results in a minute. Can I have the next slide, please? So for the constitutional results, these are all the results and the percentages of incorrect results. And you can see that there are some where it's a bit high, like at 60% for case 32, which actually was a inversion X with 48 chromosomes and an extra 21. And that was rather unfortunate that this result was not correctly given the right answer. What's more worrying, although I would say all of them are slightly worrying, obviously, as an ISCN chair, is that cases 5 and 12 were normal, and yet 25% got it incorrect. I do hope that going forward, that people have understood that for constitutional, where the main error was that they were putting cell numbers in, is you do not put cell numbers in for constitutional results unless it's mosaic. And that was the primary reason. It wasn't the only unfortunate error that was made, but it was the primary one. And also to make sure when you are submitting, you get the right sex, because that was also another error that was being made. So for a female, people were writing it down, the correct answer was male. Well, if we actually said it was a female and the karyotype result was for a normal female, you do need to put 46XX. Next slide, please, for the neoplasia. So you can see straight away that there was one result that everybody got incorrect, which was case 37. And that is the correct answer that I've given there. You do need to put 3N in because you need to say how you're writing this, whether you're writing it according to triploid or to tetraploid, and 81 would normally be reported to tetraploid, and so you need to report 3N in. Case 7, we got 100% correct, which was fantastic for a monosomy 7 and a trisomy 8, and so the chromosome number is still 46. And I'm pleased to say that for cases 16 and 27, there were fewer errors, but there was still about 15% of errors. You do need to put the cell numbers in for neoplasia, and there were a few centres that forgot to put cell numbers in, even for a normal result. So it's just to remind you, for neoplasia, you always put cell numbers in, whether it's normal or you've got clones. For constitutional, you only put cell numbers in if it's mosaic. And on the next slide, I've listed some of the common ISCN errors. Cell numbers, as I mentioned, incorrectly given for non-mosaic constitutional results, not including cell numbers when you have got a mosaic constitutional result, and cell numbers must be given for neoplasia, whether it's mosaic or non-mosaic. Punctuation, separate cell lines must be separated by slash, not semicolon. There's always a comma after 46 and after the sex chromosomes. If you've got an additional isochromosome or marker, then you need a plus before it. And remember to use DMAT when it's an unbalanced segregant of a maternal or paternal rearrangement. And again, that was a mistake that was commonly made. For monosomy X, you do need to put minus X, and for an additional sex chromosome plus X or plus Y if it happened to be an additional Y chromosome. Other errors we found were incorrect breakpoints, incorrect order of the abnormalities, it's X, then Y, and then all the autosomes, incorrect order of cell lines, and the incorrect sex. So when you've finished doing Genie, if you haven't tried it, you won't have seen this before. So can I have the next slide, please? So you get, and the next, can you move on? There we are. So you immediately get a get real-time feedback, got a certificate of participation, and the next slide, it will show you if you've got them all right. If you've got them wrong, you get a red cross, and it will tell you what the correct result should be. So you'll get the education as a consequence of that. So let's move on to the next slide, please. And to the next slide. So what are the big changes? I've tried to summarize these very briefly. There are no spaces now except between technique and genome build use and the results. So where I put the red is where you actually need now to put a space where there wasn't before. There's no space before you put homozygous if there's a number there. And obviously the extended form for the array, there's no change at all. And you do need a space between two symbols or abbreviations, for instance, pseudodicentric, and you need that before concept or mosaic or chimera. So that's exactly the same, these last two, as you had in 2020. Can I have the next slide, please? What we have done is we have just made it clearer what the different formats are, depending on the technique used and the structural information given by the assay. So for some things like genome mapping and sequencing, you may use either karyotype or microarray format, depending on what information you get given. So for the karyotype analysis format, you can use a short or detailed form as in 2020, and there is also an abbreviated form which is used for enumeration of fish probes. For instance, when you've got a monosomy 7 and a normal 7, you've got mosaicism for monosomy 7, and you would write it like this in the abbreviated form. Because it's ish, you do not put 20/35. You have to write the normal cell line as well. With a microarray format, you've got the abbreviated, short and extended system. They were always there, it's just it wasn't clear perhaps what they were. So the abbreviated system is like you have for a common aneuploidy or for a normal result. The short system is where you just put the flanking nucleotides that are lost or gained, and the extended one is where you include the flanking nucleotides which are normal. Can I have the next slide, please? So there has been some clarification, partly through EQA and partly through some of the submissions we got as an ISCN committee. We've put all the structural chromosome examples into one chapter. We've put some rules down which you sequentially apply for the order of cell lines when they have the same cell numbers. Usually it's largest first for constitutional and the normals always go last. For undisclosed sex, we've changed it because somebody said if you put query after X, are you querying that you mean an X? And so we've changed it to U, which replaces XX or XY. For neoplasia, it's not the largest first, it goes in increasing complexity, but when you have the same complexity, then the sequential rules will apply again. And again, another query we had was about what bands do you put in for recurrent translocations, and you put what you see on down the microscope, so it's the resolution seen, and it is not adjusted to the gene location. So if you saw an 821 at 400 base pairs per haploid set, these were the breakpoints, and at 700 it will be this. You do not put the gene locations in, you put what you actually see, because people will. The main concern was that people will misinterpret it, that you actually have seen it at that resolution. There's clarification of the counting chromosome abnormalities tables with some examples, because unfortunately there was a paper by a French group which had misunderstood the actual table in ISCN 2020. We've put more composite karyotype examples in. We've put some figures in for divergent clonal evolution and unrelated clonal evolution to help you sort of work out how you work out which clone goes first and what order. And we've revised the gene fusion nomenclature. Can I have the next slide, please? So for the fish, the multiplication sign is now outside the brackets unless you've got different signal patterns. So it's consistent now with RSA and array. So this was ISCN 2020, and this is now ISCN 2024. You've got space, and the ×2 is outside the brackets. When it's the different signals, they're obviously both inside the brackets. And in this particular case, this one is outside the brackets. Next slide, please. For normal results, they are given according to the probe sets in all situations, whereas before you could put them all together, now you have to keep them according to the probe sets. And don't forget, if you've got a normal ATM-TP53, you need to put the cell numbers here, not just at the end, because you're talking about the individual probe sets. And we've removed the detailed system for all normal results, so you can only use the short system. And again, this would be an example for the ISCN 2024. Can I have the next slide, please? What we did notice was there wasn't any consistency really, or there was some inconsistency, I should say, in one particular case, when you've got a loss or a gain of the same breakpoint. If it's karyotype-like ISCN that you're using, then you have to put both breakpoints in to show that it's interstitial, not terminal. With array, you only put it in once, or for sequencing or OGM or RSA, if you're using that format, if you're using the array format, because the nucleotides show you that it's interstitial. Can I have the next slide, please? That figure, by the way, is in the book. So as I said, for constitutional, for mosaicism, you've got a mixed cell population, largest cell line goes first, except for normals, which always go last in all situations. For clones, the least complex acquired clone is listed first. The least complex acquired clone is listed first. The individual neoplastic clones, and if present, a constitutional line, are listed in this following order. So the abnormal clone with the acquired abnormality is listed first. So this is the -21. Then it's the cell line with the constitutional abnormality. So with this one plus trisomy 21, constitutional, and then the normal cell line last. For both mosaicism and in neoplasia, where you've got clones, the normal cell line is always listed last. There is a small change in the rules with nukish, where the normal is in the denominator. But for the sex chromosome determination, you put it last. So in sex determination, in this particular case, because that's a normal male, you do put it last, and these will all add up to 50. And it's just so you know, because if you have this where you've got a monosomy X, here you've got an XYY, you don't actually know what the normal cell line is, and so you put it in. The sequential order rule applies for equal complexity of clones or the same cell numbers in constitutional, and there are examples in chapter 4 given for these sequential rule order for both cell lines, unrelated and related neoplastic clones. Can I have the next slide, please? So to indicate a mixed cell population where you can't precisely quantify it, which does happen, you can use now a question mark. So if you can't quantify it, you know it's greater than 2, but you don't know by how much because you can't quantify it, you can use question mark. This star, by the way, is in these examples, is just to show these are examples published by courtesy of CARGA. They're not part of the ISCN. If it's too large to enumerate, again, you can use a question mark, say, for instance, in amplification. So in this particular case, you've got amplification of MMIC, and you've got question mark here as to how much is amplified. Here you can actually quantify that you've got 80% with trisomy 8 and 80% with 4 copies of chromosome 22. If you've got multiple clones, it's optional whether you add the 1 when there's 100% present, but you need to add it obviously if there's less. There's a space there that shouldn't be there, I might add. So if you've got, in this particular case, 50% have got a loss of this particular region in 13. If you leave it here just with, as written here, people might misread it to imply this is also at 0.5, which is why we suggest you do actually put the 1 in. It adds for clarity and it stops misinterpretation. The proportion is not adjusted for the tumour load. Can I have the next slide, please? We've got some new acronyms for uniparental disomy, detected molecularly, UMAT and UPAT. There's an example of a tabulated array and also a genome mapping tabulation. There are chimer examples and including neoplasia post-transplant chimerism. We've got complex array results showing different types of chroma anagenesis, chromothripsis and chroma anasynthesis. And we've said that you can put the normal X and Y in an array ISCN if it gives clarity on the gender. Have the next slide. So for targeted analysis, other than FISH, where you use the acronyms nukish and ISH, you use the acronym RSA. And it only describes the normal and abnormal results for that specific chromosome or region tested. And we now include in this chapter, in chapter 10, is chromosome analysis when the full chromosome analysis is not undertaken, or say, for instance, you're just checking to see whether or not they've got a parental abnormality, or a targeted microRNA analysis where you're looking at specific chromosome or region. Can I have the next slide, please? So For targeted chromosome analysis, for numerical or structural abnormalities, for balanced structural chromosome complements where the homologs are apparently normal, the abbreviated or the short system can use, which is analogous to the full karyotyping, but the short system includes the breakpoints. And so you've got a normal result. Say you're looking for 1122 in the parents, that's a normal result. This is looking for, say, for instance, a trisomy 21, and it's an abbreviated form where you've got normal and you've got the trisomy. You do not put 25/30. And for the short system, you put the breakpoints in. And if you're looking for a 922, you put in the breakpoints. If you've got a non-rearranged homolog that's been lost, then the short system is used with the number of homologs described as 0. That's so you understand how many chromosomes you've got. Otherwise, you don't know if you've got 2 normal chromosomes on top of the ISO, or you've got 1, or you've got none. And the same system here, where you'd use this for an 821, where you've got 1 normal 8, but you haven't got a normal 21. If it's unbalanced structural, only the short system is used, and the normal chromosomes, again, are listed in the ISCN nomenclature. Next slide, please. This is for targeted array. It's very similar. There are some slight differences. So this would be a normal result for a targeted analysis of chromosome 22. If you're looking specifically for the critical region on chromosome 22, you only put this breakpoint in once, because you can tell from nucleotides whether, because it's a normal result, sorry. But if it was abnormal, you'd only put it in once, because you could tell from nucleotides that it was interstitial. You put the nucleotides times 2. So if you've got loss of a deleted of the 22q11.2 region, if you use abbreviated form, you would write the breakpoints times 1. In this particular case, you would just write breakpoints once, because you can tell from here that it's interstitial. And if you do the extended, then you've got the normal flanking nucleotides. Please note that in the extended system, as was in 2020, you do not put commas, because you start putting commas in, you suddenly don't know what this number is. Can I have the next slide, please? So for the sequencing, we have changed the terminology and made it much more clear. So we've given you ISCN examples for different platforms and bioinformatic algorithms. There's more normal aneuploidy mosaic and complex examples. And we've also said where you have to use both, because they give HGVS and ISCN because they give different information, whether either because they give exactly the same, or where ISCN is actually more informative, for instance, for the inheritance patterns, because you can't give that in HGVS. And again, as I mentioned before, the breakpoints are only listed once, and this was a discrepancy before in the ISCN part of the nomenclature. We've also put in normals. You will obviously have to put some limitations and explain what the resolution that you're working to, but you can in ISCN put a normal result for the sequencing. And for an abnormal, here is a situation where you can use either, because they both give the same information. Maybe I'm biased, but I personally think that is a lot simpler than using HGVS, because in HGVS you can't say whether you've lost the X or the Y, and so you have to describe both. This little mark here means all. Next slide, please. So for genome mapping, very similar to array, and exactly the same. And when it's not disclosed, again, in with array, you don't use U, you just put 1 to 22, you just don't put sex chromosomes in. And for aneuploidy, exactly the same as for array, you're just using the OGM abbreviation. It's exactly the same for constitutional neoplasia. There's no differences, and it can be used for obviously multiple different abnormalities. Can I have the next slide, please? When it comes to genome mapping, you can use karyotype or microarray format. You use the karyotype format if the structural nature of the abnormality is apparent, in which case you do not use copy number. If you use the microarray format, then you do use copy number. The platform, labeling strategy, and bioinformatics pipeline are given in the narrative, not in ISCN. If you have, say, a neoplasm, multiple abnormalities, you can present these in a table. The VAF can be given in the ISCN, as can the gene involved if it is relevant. And the nucleotides, do remember, indicate the extent of the fragment determined by the positions of the labeled DNA motif within the variant region. So they're not necessarily those of the actual breakpoints. It's a limitation of the test. Can I have the next slide, please? So for a deletion, you have you can use the array format with the ×1, and you can use the short format or the extended format where you've got the flanking nucleotides. You could also use the karyotype format if you can actually work out what the structural rearrangement is from the technology. And so because using the karyotype format, the breakpoints come in twice. You put the flanking nucleotides, but you do not put ×1 because you're using the karyotype format. If there is a clinically relevant gene, you put it before the number, before the copy number. And if you have mosaicism, that goes after the copy number. And if you've got a constitutional abnormality, this goes again before the copy number. Can I have the next slide, please? If you've got deletions involving sex chromosomes, as I mentioned earlier, you can put the normal sex chromosomes in to give clarity, and they were listed first because you've got an abnormality of the X chromosome, which is the same reasoning you had in the karyotype and the in the karyotype format. If it's inherited, the MAT or DMAT, if it was a DMAT, would go after, with no space, after the copy number. And in this particular case, this is one where you've actually, this is the microarray format, where you've got an interstitial deletion on Q225. If it was inherited, then because using the karyotype format, you put the breakpoint in twice and you put MAT afterwards and you don't have the copy number. Have the next slide, please. Obviously, genome mapping is great for translocations. And you don't always know exactly where the breakpoint is. And so you can put a range of breakpoints which you separate by tilde. So this is the breakpoint on 6Q21, and this is a breakpoint area on 7Q32.1. If you've got a three-way translocation, for instance, involving a fusion gene, this just shows chromosomes 6, 12, and 21 in the circus plot. You put the breakpoints. There's this karyotype format. This is the range of breakpoints for each of the chromosomes 6, 12, and 21. Then you can put, you've got an ETV6 RUNX1 fusion, and you can put the VAF after that. Because you're using the karyotype format, you do not put copy number. Can I have the next slide, please? So for inversions, we've had to make some extra nomenclature up. They may appear identical to a translocation between homologs using OGM. And so we had to develop a specific nomenclature. If the mechanism isn't apparent because technology doesn't allow the homologous 16s, for instance, to be distinguished, you don't know if you've got an inversion 16 or you've got a T16-16, then you have to, if you use a karyotype format, you have to put both options there, and it's all part of the same ISCN nomenclature with an or and space either side of or. There's no carriage return. Alternatively, without giving the mechanism, you could just report there is a fusion between 16 and 16. These are the breakpoints that are fused together. These are the nucleotides, and this is the gene fusion. So you've got both options. Have the next slide, please. And for ring chromosomes, very similar to, in this part, to gene karyotyping. And again, you're talking about a fusion between the two ends, and there is a loss of material. And so you've got fusion, and these are the breakpoints that have joined together. And this is another one where the breakpoints perhaps are not so clear. Now we're on to the quiz. Have the next slide? Yes, we're on to the quiz, folks. So I'll give you this one really quick one. Some of them you have a bit longer to think about, but this one is very straightforward one. What percentage of laboratories that submitted the correct ISCN for the constitutional Genie module?
Ross, do we need to just give some instructions of how to actually vote? So
I
if you,
assume they can vote online, and you can see which of the options you've got.
Yeah, so if you go to the user bar and go to the section that says polls and drop down that menu using the triangle on the left hand side, you actually have the 4 different options there. So you can see people starting to vote now, so you can select the one that you think is correct.
Well, I think we will close the poll.
Yeah.
And most people thought it was A. Can I have the next slide, please? If most people, which
So
is
we
correct.
had 82% saying A, 10% saying B, 2% C, and 5% D.
So I'm pleased to say most people got this right. I would still love to see 90%. Yeah, that's where we're at. So let's move on to the next question. Now we're going on to actually ISCN. So these are ones that either come out of EQA where people got them wrong, or they've come out of Genie. And so I thought it'd be good ones to test your knowledge on. So you've got a male karyotype with an inverted duplication of chromosome A, I've shown you it here on the right here, involving bands 8p23.3 and 8p21 in 20 metaphases. So which is the correct ISCN? We're talking about constitutional here. Do you want to go to the poll?
Okay, I think people have stopped voting now, Roz.
Yeah, stop poll. Can you have the next slide, please? The correct answer is actually A. Most people had it as B. The breakpoints are inverted. And there's no semicolon for duplication, and the chromosome count is incorrect for D, but nobody got, or only 2% got D. Next slide, please. So you've got female karyotype, 46 chromosomes, 1 normal X, and an isochromosome in 10 metaphases. There are therefore 3 copies of the long arm of the X chromosome. So which is the correct ISCN? I think we'll close the vote. Next slide, please. Well done, everybody. Vast majority of you got it right. A is wrong because there's no cell numbers given for constitutional, and D is incorrect because that would indicate 3 X chromosomes. B is incorrect because it was an Is chromosome with a P arm, and it was actually a Q arm that was an Is chromosome. So the next question, please. Next slide. Here we are. A female karyotype with trisomy 21, 3 X chromosomes, one of which is involved and has an inversion with the breakpoints 8p21 and 8q23 and 30 metaphases. So which is the correct ISCN? Oh, we're struggling with this one. Right, let's close the poll. So now next slide, please. The correct answer is C. A is incorrect, which most of you, the majority of you got, but there was 30% of you thought there was a plus. There's no plus in constitutional sex chromosome abnormalities. You just put inversion, and B would make, you've got extra X chromosomes, so that would make it 49 chromosomes. So once you've got an inversion or an ISA, you don't repeat the X chromosome in the first part of the sex chromosome constitutional. Next slide, please. So this is an neoplasia one. You've got 30 metaphases from a female patient with AML. 25 showed acquired loss of an X chromosome. 5 showed a normal female karyotype, which is the correct ISCN. Can I have the poll, please? I think we can close the poll.
Oh, B's well out there, Ross.
Yeah. It's interesting how for neoplasia, whether it's the individuals who are doing it, neoplasia gets answered much better than constitutional, but that is correct. And the A is wrong, just got cell numbers the wrong way around. Can I have the next question, please? So this is 10 metaphases, male patient, neoplasia again, with an interstitial deletion of chromosome 20. And the breakpoints are 20q11.2-13.1 and 20q13.3. And that is actually a breakpoint in ISCN. Do you want to open the poll? Let's close the poll. Can I have the next slide, please? The actual result is A, which was the second most popular result. B is incorrect because it would be correct if it was constitutional, but you need cell numbers. C is incorrect because the breakpoint doesn't have you have to put all three parts of the breakpoint in. So you have to put this part of the breakpoint and this part of the breakpoint. And this is incorrect because you put a Q in here and the breakpoint does not have a Q there. If you go and look at the ideograms, you'll see it. There's a couple of breakpoints that are just like that at low resolution. Next question. So this is 20 metaphases analysed for a male patient. Again, neoplasia, 2 normal copies of chromosome 8, as well as a translocation between 821. And then there's a normal carrier type as well. So which is the correct ISCN? We have the poll, please. Half of you voted, so let's close the poll. And the majority of you got the correct answer, which is brilliant. Can we have the next slide? Next slide. That's it. So those of you who got a, oh, that one. Can we go back one slide? There we are. So those of you who chose A, you forgot we're talking about Q arms, not P arms. For B, it's numerical before structural for the same chromosome, so +8 always goes before the T. And then C, which only if 1 or 2 of you got wrong, normal cells always go last. Next question. So you've got prenatal, maternally inherited, T1122. What does it want to know? The sex. So which of these, A, B, or C, is correct? We have the poll, please. Thank you. Oh, we can close the poll. Well done, everybody. Can you have the next slide, please? 92% of you got it right. It's a figure I love to see. So they would have been right in ISCN, but there's no space before Matt after the bracket, and B is ISCN 2020. Can we have the next question? We've got 2 more to go, and then we'll do questions quickly. We'll just be a bit over time. So you've got a mosaic result, 3 cell lines, all have 15 metaphases. Which is the correct order? And this is a change in ISCN 2020, which if you've looked at it, you'll get this correct. And if you haven't looked at the book, you might make a mistake. Right, can we close the poll? Go on, next slide, please. So the answer I've been given is as correct as B, which is 64%. And that basically is ISCN 2020. And the difference is we had a difference in the book in 2020 where for neoplasia case, it was gains before losses. For constitutional, it was losses before gains. So we've made it consistent. It's gains before losses. So answer C is correct. Those of you did what I think probably one individual put A, normal cell lines always go last. And then quick one for 2 more quick questions. 10, targeted chromosome analysis for chromosome 8 only, non-mosaic, non-disjunctional trisomy 8 in 10 out of 30 metaphases. You could actually, this because it's mosaicism, could be either a constitutional neoplasia case actually. So if you put the poll through, let's see what answers you choose. Can we close the poll? Next slide, please. Well done. But majority of you got the right answer. For the few of you showed A, the denominator is not used in RSA, it's only used in UKish. For B, normal cell line is always listed last. If nothing else, I hope you get this message. And for C, this is where it's a bit tricky. This is a targeted chromosome analysis. C will be correct if it's a targeted microarray analysis. Next slide, please. It's the last one. So this is genome mapping, and you've got 22q11.2 deletion. Which of these is incorrect? There's only one that's incorrect. 3 are correct for ISCN 2024. Can we have the poll, please?
Just to say for this one, D doesn't appear because it's too many characters for the slide deck voting system.
Okay. So 2 are correct and 1 is incorrect. Right, let's close the poll. Well done, everybody. Next slide, please. Yeah. The one that is incorrect is B, because it implies that the whole of chromosome 22 was analysed. The others actually are all correct. A is the abbreviated form, C is the short form, and D is the extended form. So have we got any questions?
Just a couple for you, Roz. One about, can we use the online ISCN login for multiple users?
I don't see any reason why you can't. I don't know. I've never tried it. Try it, see if it works.
Yep.
I'm not sure if it picks up whether you, I mean, I would have thought so because I've worked in different locations and it lets me get in, but honestly, I've never tried it.
Okay, thank you. What about the slides for, in your neoplasia slide earlier, you showed an acquired loss of X in a female. So you had it as 45, XX, minus X. Is that a typo or is that correct ISCN?
Oh, hang on. Where is it?
So it's the neoplasia slide. So an acquired loss of X in a female.
45, X minus X.
So just one X rather than
Yes.
2 X's.
Yes.
Okay. Okay, thank you.
Let me just check. You've got me worried now.
You see, even the experts have to still look up the book every now and again.
I mean, one thing I didn't say was, look, I make mistakes. You know, doesn't make me perfect. Is there another question while I look this up?
Yeah, there's quite a few others coming in. Let me just expand.
No, it's 45X, minus X. It's on page 39 in section 5.3.1.2.
Right, there we go. Right.
Sorry, I had to double check myself.
That's fine, we believe you. Imagine a situation for a potential normal embryo, so XX or XY, analysed by shallow sequencing, and that we're not allowed to report the sex. We could actually put 46, U. However, with our technique, we're not able to distinguish a truly euploid from a triploid. So we can't see variations on ploidy. Can we use the 46, U in these situations, even though we don't know the correct ploidy?
No, because you're using sequencing, you're doing shallow sequencing, so you should be using the sequencing ISCN, so you should use SSEQ, and then use the, effectively, if you don't want to say what the sex is, do it such that if you're looking at all the chromosomes, you do SSEQ, space, open brackets, 1 to 22, close brackets.
Okay, thank you very much. When do we use DMAT versus MAT?
Right. Think of a translocation. If mother has a translocation, it's and she's inherited from her mother, it's MAT. If it goes to the child in the same form, it's MAT. If, however, you get a derivative, so it's unbalanced, then it becomes DMAT because it's derived from the maternal rearrangement. Does that make sense? We have written it in the book, and we've given some examples.
Right.
Get back to me if not.
Excellent. Melody, who's part of the team at GenQA, one of the deputy directors, has said, can we promote the ISCN EQA round 2, where you can practice the RSA nomenclature for targeted karyotyping and targeted array tests? So if you're not already registered, then please sign up to the lab-based ISCN round 2, which I think goes out in the autumn time. And we've got one final one in here.
Pilot,
also,
so you just get feedback. You won't get poor performance.
That's great. We've also got one of the participants says that we have potentially an error in the Genie question number 37, which is why everyone got it wrong. Might be due to the correct answer is not active to be selected. So that's quite good. If we could get a screenshot of that, yeah, we'll investigate, which is good news that that's the reason why maybe
Thank you for
everybody,
the feedback.
Yeah, absolutely brilliant. Right, we've got 2 more and then we're going to close the session. So what do you write if you're analysing a leukemia sample and you find XXY but are not 100% sure whether it's constitutional or acquired?
Right, well, there's 2 ways of doing it. You can put query C, but you put XXY query C, or you write the 2 options out, because I've had this discussion already with somebody. I'm trying to think where it was. I think it's in chapter 6, which is the neoplasia chapter. And you can put the 2 options. So you can write it as 47XXY, and then whatever abnormalities you've got. Then put or, and then you write 47XY plus X, plus all the abnormalities. So there are 2 options you've got.
The 2 option, okay.
Yeah,
brilliant.
you either put both options in, or you put query C after XXY. You do not write XY plus X query C, because that's, you're actually saying you're saying you've made a decision, it's acquired if you do that.
Okay, thanks, Ros. One final one. So if if rearrangement is identified by FISH, are we able to use 700 base pair HS ISCN for karyotyping?
You have to go back and look at the karyotype to see whether you can visually see it. If you can see it, you can change the breakpoints. If you can't see it, you just change the breakpoints on the FISH. It is what you actually can see, because otherwise you're implying it's there when you actually can't see it. It's like saying the 1221 is visible when you can see it on FISH, but it's actually cryptic and you can't see it on G-banding.
Great, thank you very much. So Ros, thank you, incredibly brilliant educational webinar as always. We will get our participants will get a survey sent to you, so thank you for joining the webinar, and we really would like to hear your feedback, because we do focus these webinars on areas that you wish us to specifically address, so we do need your feedback to be able to do that. Shortly the webinar will be available. It's about 1 month's time to view on our GenQA YouTube channel, so if you're not already subscribed to, there's a lot of many webinars there with some great educational content, and you will be sent a participation certificate as well. We haven't got a date yet for our next focus on topics, so please sign up to a newsletter and you'll make sure that you get access to the latest information as and when the webinars are arranged. So we'll just finally, thank you very much to Angie that manages Jeannie, to Melody and Mark as well, who support in the ISCN EQAs at GenQA, to Emma for running this lovely webinar with our fancy polls. That was great fun, and Ros, of course, you as well. Thank you, and thank you to you participants for listening. Thank you. Bye-bye.
Bye.